All right, everybody, we can get started. I'm going to give a quick update or a quick introduction about the organization to get us started here. And hopefully we get a couple more people shuffling in as that happens. My name is Ted. I serve as the Lower Susquehanna River Keeper. Thanks, everyone, for joining tonight. We're going to be going over our bacteria monitoring program tonight. With me is
Aaron.
Aaron Dixon's our program coordinator. He's going to be going through a lot of the detail of the program. But just to start us out, give everyone just a quick introduction as an organization. So our mission, we've been here, actually, this is our 20th year. We're celebrating 20 years this year. We were formed in 2006. We are a licensed waterkeeper organization. We received our license in 2007 from Waterkeeper Alliance. And our mission is to improve the ecological health of the river. And we do that through education, through research, through advocacy, and insistence upon compliance with the law. And so if you break that up, our work surrounds and revolves around three major buckets. So advocacy and enforcement, community science, and education and stewardship. Our territory as a waterkeeper organization, again, we monitor and patrol for pollution in the lower Susquehanna River watershed, and that's from Selinsgrove, Pennsylvania, all the way down to Haverty Grace, Maryland. It also includes the Juniata watershed, so you can see there on the western side of the map, all the way from Raystown Lake to Duncannon. So what do? We monitor for pollution in the river and all of its tributaries by patrolling, investigating, collecting data. We insist that environmental laws are enforced by regulators, and sometimes when state and federal agencies fail to do their job, it's waterkeepers that step in and enforce the law. We use science, so we build coalitions and we file reviews to update our laws and regulations to improve water quality. And we also educate our lawmakers and youth and adults on the river's ecosystem and a lot of the issues that it faces. And we also organize activities like volunteer cleanups and programs such as this, bacteria monitoring. And we also love to bring people to the river. So we host a lot of different events and other types of programs that bring people just a little bit closer to the river. So we have paddling programs through the summertime. And we also have cleanups all throughout the year, and some other events we have here locally here in Wrightsville, where we have a park that's literally right across the street from our office. So that's a very short introduction for everyone tonight, but that's not the reason why we're here. Is to talk about all what we do. We really want to get into bacteria monitoring, and Aaron's gonna give a quick update about our other programs under our Water Watchdogs programs. So bacteria is one of them, and Aaron's going to go through some of the rest here.
Yeah, so like Ted said, we can't really talk about bacteria monitoring without talking about which umbrella of programs that falls under, which would be our Water Watchdogs programming here at LSRA. Which encompasses bacteria monitoring, but it also holds microplastics monitoring and smallmouth bass monitoring. To talk just briefly on the other programs, the other microplastics monitoring and smallmouth bass monitoring are currently being re-envisioned, redeveloped in a way. With our microplastics monitoring, it's being refocused. Science and Policy Committee, which is chaired by Mitch, who's on our board. The idea behind that monitoring program is taking water samples in and around wastewater treatment plants throughout our watershed and collecting those samples and then analyzing them through a system that looks for microplastics, essentially. The other program, so smallmouth bass monitoring, is in the late phases of being developed, re-envisioned as well. We have been working with for, I think since 2020, 2021 with USGS fish biologist Vicki Blazer, who's been extensively researching uh, pollution and how it affects smallmouth bass health within the Susquehanna watershed and other watersheds as well that flow into the Chesapeake Bay. And currently, the way that app or the program will look is through an app that we're developing with Chesapeake Commons. And it'll be a way for fishermen to, when they're out there angling and catching fish, if they notice something off with their fish or their catch, they can then take an image of it and then report it through the app by somewhat self-diagnosing their fish that they've caught with help through the app. And yeah, our goal is to hopefully have that out and ready by this summer or next fall. And then that brings us to our bacteria monitoring program, which everyone here is here for. The layout of what I'm going to discuss for the program is going to be the what and why of monitoring, where and when do we monitor, how do we monitor for bacteria, what happens with those results that we collect, and then where we are monitoring in 2026. And then at the end, this is the first step of learning a little bit about the program, seeing what's all involved. It's a little more intensive than some other volunteer programs out there. So we'll have trainings and I'll talk about that towards the end of the session here. So first, what are we monitoring? What are we monitoring for? So we're looking for bacteria. That bacteria is total coliforms, which are bacteria that are found in the soil, water, and influenced by surface water, and it originates in human or animal waste. The specific type of coliform that we're looking for is E. coli. So that is a bacteria, like I said, with coliforms, it originates in the intestines of warm-blooded mammals. And how we're monitoring, like determining the presence of it is through what's called the most probable number of E. coli colony forming units. And so we're monitoring for E. coli. Why? Why are we monitoring for E. coli? So it's a good indicator species of fecal contamination in our waterways through the work of TED. This program essentially originated in our organization through the testing of CSOs up in Harrisburg. If you look in the image of Ted there, he's holding his nose, and he's holding his nose because that is a discharge from a CSO that is spewing raw sewage into into a tributary that flows right into the river up in Harrisburg. The other way that raw sewage or fecal contamination can enter our waterways is through livestock. In our area, in Pennsylvania, there's no regulations on exclusion fencing for livestock. So a lot of farmers in our area allow their livestock to have free access to the waterways that are on their property. It's an easy way for them to allow drinking water for their cattle, but it opens the door for that livestock to go to the bathroom and poop in the water that flows through the farm there. And then in that below image there below Ted, you can see from a drone image in York County, that's some cattle that have free access to a tributary to the Susquehanna on somebody's farm. Why do we monitor for E. coli? What's the big deal with fecal contamination? Well, with E. coli, it's known to cause illnesses. It can cause traveler's diarrhea, vomiting, cramping, UTIs, pneumonia, sepsis, fever, and in bad, worst case scenarios, hemolytic uremic syndrome. Where did we monitor this past season? So for the whole season, we've monitored 22 sites. In 2025, in York County, we've monitored Wrightsville, Lake Clark, which is a lake that's created by Safe Harbor Dam on the river, and Lake Audra, which is created by Holtwood Dam on the Susquehanna River. We also monitored Yellow Breeches, Cadorus Creek, and Muddy Creek. In Lancaster County, we had 9 sites. We monitored in Columbia on the river, Lake Clark, again, Lake Aldred on the Lancaster side, Conestoga River, Little Conestoga Creek, and Peckway Creek. And then in Dauphin County, we monitored 3 sites on Swatara Creek and then the Susquehanna River at Harrisburg City Island. When do we monitor? So this is going to be one of the key factors for most people and being able to have the availability to take part in this program. The bacteria monitoring does occur during Thursdays and Fridays during the daytime hours, working hours. So if you're unable to, if you're working full-time job and unable to take off those days throughout the season, this might be a confliction for some of you, but just want to be as transparent as possible with that information. The way that we monitor, so it's all scheduled around what's considered the swimming season from Memorial Day to Labor Day, which is acknowledged by EPA and PADEP. This year, those dates will range from May to September. It's going to be a 16-week program, which is new for us. It's always been a 15-week program, but this year there's 16 weeks for the swimming season. If there's ever a holiday, it doesn't happen. It actually didn't work out this year where we didn't have to add an extra day of sampling. But if there's a holiday that falls in the middle of the week, we'll try to go out 2 days prior to that major holiday and collect samples. But it works out well this year and don't have to worry about a middle of the week sampling. And then we'll results on Fridays, which is 1 day before the weekend. The goal of this program is to bring awareness to water users, and most of the folks that use our waterways, it occurs on Fridays and weekends. So we want to have those test results out to the public so they can be aware of the conditions of the water before those heavy usage days. Where are we hoping to monitor in 2026? So we are going to be removing one of our test locations. That's going to be in Kleins Run Park in York County. The reason for removing that is, one, to increase our capacity to monitor other areas that need to be monitored. But it also is really, really close to another testing location that we feel confident in. The test results that come back between both locations generally are about the same. So it makes sense for us to utilize those resources a little bit better and more strategic. This year, where we like to grow, it's not definite. It's going to be very based on what kind of volunteer support that we'll have this year. But where we'd like to go is Conestoga Creek. There's going to be 2 locations yet to be announced there, but hopefully 2 locations for Conestoga Creek. And then Goldsboro at the PA Fish and Boat Commission launch. And then in Lancaster County, we'd like to add 2 more locations there. One at the Susquehanna River at Falmouth, at the Falmouth Trailhead parking location, the river access there, and then one at Conestoga River Park on Conestoga River. And for Dauphin County, we'd like to add Susquehanna River at Steelton, at the Steelton boat launch there. So we covered the what and why and where. What what's more involved is how. How do we monitor? So everybody here, or those individuals who want to volunteer, there's going to be in-person trainings. And those trainings are going to be in full detail. We're going to run through the manual. We're going to practice collecting water samples here at our headquarters. And then we're going to have a chance to run through all of the data collection that's going to be collected at the location. So all the field collection, which includes, which I'll cover here in a little bit. And then the trainings, what they provide is just skills through practice, and then you'll get to practice with your potential teammates as well. So you'll get to familiarize yourself with each other and just really hone in those practices. The water collection. So what we'll be practicing for the water collection portion will be collecting the water sample. There is a specific way of how to collect that water sample. So we'll practice that. We're also going to be collecting other data like weather, rainfall, 24 hours and 48-hour rainfall, flow, turbidity, and temperature data as well. Each team is then, after you collect those water samples, each team is then going to be responsible for transporting those samples. This year, for some locations, there's going to be a little bit more effort for figuring out logistics. So either maybe a meeting location if there's for those out stretches, further away from our headquarters, if it makes sense for some teams to connect with each other and just pass off those samples to make that transport a little bit easier. But in the end, each team is going to be responsible for those samples to be transported here. And then when you drop off the samples, those samples are then going to be held on ice. They can be held on ice for up to 8 hours, and then we'll process them. We're going to process them with the Color 18 method, which is manufactured by IDEX. That method is a EPA certified method for analyzing E. coli. It's considered a substrate test, which we'll go into more details of how that happens and works during the trainings. But it essentially is just an enzyme detection test that when it reacts, when there's certain substrates in the reagent that we mix into our water sample, it's going to react with certain enzymes that are produced by E. coli and total coliforms, and then produce a physical reaction for us. Yeah, processing those samples, we're just going to practice aseptic technique with being as clean as possible. We do have a lab here that is going to allow that to be pretty easy to do, a nice sanitized area. And then, yeah, those samples are essentially the samples that we're going to collect are 100 milliliters worth of a water sample. So we'll make sure that those water samples are dropped down to that 100 milliliter line. Sometimes you can overfill those water samples, but we'll get into those techniques and figure out how get as close to that line as possible when collecting that. The samples are then processed with that method, that collared 18 method, and then are placed in our incubator for 18 to 22 hours. And then that allows, well, we'll go to the next slide, and I can show you guys the little bit more of the science behind the reagent here. So the substrate is essentially going to be, these images here are demonstrating essentially the substrate that is in those reagent packets, which is in those granulars. And this just shows that when it finds a certain enzyme that is produced by the bacteria, it'll then form and then cleave. And then when it cleaves, if it's a coliform, it'll show a visible yellow there on the left. And then if it is E. coli, it'll cleave off off the substrate, and then a piece of that substrate will glow. It'll be a forgive me, I'm losing my words, but it'll essentially produce, when you put the trays under a black light, it'll produce a fluorescence type color to it. So then what that does is it'll determine that what we're looking at is a coliform, and if it's yellow and then it glows blue under black light and it's E. coli, all that's doing is just making sure that we're confirmed and confident in knowing that what's in that well of the tray that I'll show you at our training is in fact E. coli in those wells. And then analyzing for those results. So just like what I was describing, we're going to be looking for that yellow, which is going to be visible to our eye without the need of tools. And then the blue as well is going to, the fluorescence will help us as well. And the way that we'll analyze is it's going to be in trays. And I'll show you those trays at the training, like I said, and then there's a method to counting those wells, and then there's going to be a table that we're going to use that will that's for determining how many positive wells we have that are confirmed to have E. coli in them, and versus negative wells as well. And this is just to kind of briefly cover what I just talked about here. So we're going to collect those water samples. We're going to record the data while we're at the location. And then we're going to come back, process those samples, incubate at 35 degrees C. And then the next day, this is a good image here of that tray. If you notice, there's some wells that are yellow. So those wells have some sort of coliform in them, but since they're not fluorescing blue, it's not E. coli. But if you look at the wells with the X's on them, that X signifies that one dash is yellow, and then the other dash is signifying that it's fluorescent there. So those are confirmed. Anything with an X is a confirmed well. And then we'll count those big wells. There's also little wells as well that's not in the image. And then we'll determine the MPN value utilizing that chart. And then what happens with those results? So we're going to, after we're analyzing, we're going to figure out those MPN values. These graphics on the right here are from 2024. And what they're essentially showing, these are the graphics that we use for all of our social media, our website, the weekly wave email that we send out. And we also use an app called Swim Guide, which our images aren't published on the app, but the app allows us to publish our results. That is a map that you can use and determine through their program whether those locations are safe for swimming or not. And then these are just site-specific images that we use. If you notice in those images, there's some yellows and reds and greens. The green means that it's a good test result. It's below the PA DEP standard for determining E. coli in the waterways. The yellow means that it's there's the yellow is just cautionary, and then the red means that it's above that 1-day testing standard, which means by PADEP's recommendation that we should not be entering the water. And what what this does for the public is it just helps them figure out, maybe their favorite watering hole isn't a good location to swim that day, but maybe they can find another location that's nearby with a green star and go there instead. Or if they're all red, then maybe it's a good idea to, just find another activity for that day. But all for the public to better understand our waterways. And with that, that's the end. So I just want to quickly talk about, I know that was a lot of information being thrown at you in the past half hour here. Our trainings, we're going to have 2 days, not 2 days of trainings, but 2 days for folks to sign up for one of those days. One of those days, I don't have the dates with me quite yet. We need to finalize on those dates. And what I plan on doing is everybody's email that RSVP'd will get a copy of the recording here. So if you want to go back and re-listen to this information, you're gonna get a copy of that, and then a PDF version of the presentation here, so you can go through and read the information again a little bit more thoroughly. And then there's going to be a couple of forms. One form will direct you to one of the days for signing up, or the other form will direct you towards the other day. The idea of having 2 days as options, one option is going to be during the day, a weekday during the day training. And then the other one is going to be a weekday evening training. So for folks who want to come during the day, that have more availability during the day, they can pick that day. And then for folks who prefer to come in the evening due to work hours, they can pick that day as well. With that, that's all that I have. If anybody has any questions, there is I know there was the chat was going off a little bit. There's also a Q&A location at the top of the screen if you guys have any questions. And then we can also, I can unmute you guys here, and if you guys would like to talk, let me. Actually, I'm blanking on how to unmute everyone. So if you guys want to raise your hand, that'll let me know if you guys want to chat and ask a question. So. And then, oh, yep. There you go, Diane, if you want to come off mute. Oh, I think you're still muted. I enabled you. I think you still have to go and hit the unmute button.
Yes, hi there. Hey, I'm just curious why the smallmouth bass was chosen, like with multiple species of fish in the Susquehanna. Why the smallmouth bass to monitor?
Yeah, so with the smallmouth bass here, let me go back to the slide here real quick. So looking at the slide, I did have some information here that I didn't cover. But through the work that Vicki Blazer has been doing, one, smallmouth bass is one of the largest fishery,
Like
one of
there's
the
a
most
lot of them?
profitable fisheries. Yeah, there's an abundance of smallmouth bass in our watershed, and in the East Coast as well. So it's a big deal as far as what anglers prefer to catch, and as a way of generating funds for state and federal use. So they're monitoring that fishery. They've also noticed studies have shown an increased rate of disease within the smallmouth bass populations. If you're looking at this image in blue, it's kind of hard to tell. But in the winter months, this is just one issue that has occurred, but that's a form of melanism in the smallmouth bass. So if you see those black blotchy areas, um, that's it's a it's
Like
a
a pigmentation thing.
it is, yeah, it's like a pigmentation in the epidermis of the fish, and it's been known to be more abundant in the winter months, through Vicky's work. There's also a lot of studies as far as through fish tissues and sampling that way. There's been PFAS that's found in the tissues of smallmouth bass fish. And if people are catching these fish, and it's a big fishery for us, some people do eat smallmouth bass. And uh, it's good to do a study on a fish that people might have to rely on as a source of
Yeah,
food
so
in
so
our
basically
watershed.
there's a lot of them out there. People are catching them, so you have more opportunity to gather information. Yeah. Yeah,
Yeah, and
thanks.
then all the information that we collect is going to be one used internally by us. If there's hot spots of one type of disease or multiple diseases in one location in the river, that could maybe help us, pinpoint the cause. Or what we'll also do is provide all of our data to the research team that Vicky manages or managed. Vicky is actually just recently retired, but her team is still active, and still studying the fish. So
Sounds good. Yeah, thanks for the extra info.
yeah, of course. I also noticed, Diane, in the chat here, you asked about CSOs, which I do want to just cover real quick. So what a CSO is a combined sewer overflow. It's where stormwater systems and sewer systems combine, and when it's dry out, they work pretty well, where they stay separate. But when there's a lot of rainfall, or in the sense of Harrisburg, because it's so big, it doesn't take a whole lot of rainfall um, to overwhelm the system. But what will happen is that rainfall will flow into the stormwater system. It'll it'll overwhelm that system, and then it'll cause it to flow into the stormwater system, overwhelm that system, and then they combine, and stormwater has a discharge, and that sewer will then enter through the stormwater discharge that will then empty into the river. So it's a very old design of managing stormwater and sewer. Um, Not every city has a CSO. There is a big push to separate CSOs and redesign those systems. But there are plenty of cities that still rely on a CSO.
Aaron,
Um,
Karen Green has her hand up.
okay.
Hi, everyone. Can you hear me?
Yes.
Yep.
Okay, great. As someone who's been a team leader for really since the beginning of the program, and we're looking to add the kind of to go in it and recruit new volunteers, would it be okay to suggest that a minimum requirement would be for folks to do the sampling on Thursday and would not necessarily have to be available for Thursday and Friday? Would that would that be okay to
Yeah.
suggest?
Yeah, so the way that and it'll be discussed more in our trainings too. Thank you for pointing that out, Karen. So as far as the bacteria monitoring program, how it operates, most of our need is for collection. So collecting those water samples on Thursdays. Whereas our lab is fairly small, so we can only hold maybe up to 10 people in our lab at a time. It's still a very big need to have help in the lab on Fridays and Thursday afternoons. But as Karen's mentioning, water collection comes first, and we're going to have interns helping us this year, hopefully, and that'll help us do the internal process of the lab. So being here at the lab, if you're further away, like Conner de Gwinnett and Yellow Breeches and Harrisburg and Swatara, it's not a mandatory thing to be present in the lab if you're out on Thursday. So whatever you have availability for is what we'll take.
Also,
Did
there
that
was a
help you?
question in.
Go ahead, Deb.
Sorry, yeah, there was a question in the chat. Will we be in a team of 3 or more in order to accommodate summer absences? So I'll let you touch on team sizes.
Yeah, so teams are I think our smallest team is a team of 4, a team of 3 or 4. And that's a pretty good size. We can probably we do have teams of 6 or 7 as well. So if we do want to make sure that we have enough coverage, because we do not want to have one person on a team and have them be responsible for 16 days during the swimming season. So if we can find at least 3 or 4 people, if not more, to be on a team, then we'll know and we'll have confidence in being able to operate that team for the season. If we have people that sign up that only one person is able to be on that team, but they really want to be involved, then we'll try to assign them to another team that's close to them as well. And yeah, so Dean Evans just mentioned about drop-off location. So that's going to be at our headquarters, and I failed to mention where our headquarters is. That is in Wrightsville, PA. It's the 338 South Front Street here in Wrightsville.
And just another point for sites. So we're going to be providing in the forms that Aaron talked about earlier. There's going to be options to choose specific sites. So all the sites that you saw here tonight, depending upon what feedback we get from our teams, you may not see all the sites you saw here tonight on the form for you to choose. So if you're here sitting here tonight, you're like, oh, I'm gonna do that site. Wait until we get out the form, and then you'll have the opportunity to see what's going to be available to choose. So you will have the opportunity to choose your most favorite location to go to, and then you'll be able to choose a second location as well. We do want to be able to spread out our volunteers evenly throughout the team. So yeah, ideally, it'd be great to have 4 to 5 people on a team. That way, each person can take a week of the month and spread it out evenly. But so those are coming within the week. We'll have those out to you, and then you can choose them where you want to go. If it doesn't work out, that there's a location that's close to you and you're not comfortable taking a further drive, that's fine. Please continue to support us. We will have, further options next year as we continue to expand this program year after year. We've been doing a very good job of doing that. So we hired Aaron in 2023, I believe. And so Aaron piloted this program with just 5 locations. And within 1 year, we expanded to 15 locations. And then in the third year, we expanded to 22 locations. We're looking to expand here to 25 or about 27 locations this year. So we're continuing to expand year after year. So stay patient with us. Again, if there's a site on here that's just not where you live or not where you can get to, we'll be again expanding as we go. So stay tuned. If what you saw here tonight doesn't suit you, and if it does suit you, great. We're going to be excited to have you join us here in 2026.
And then two more questions that just popped up. So the hours again, they are for sample collection days on those Thursdays. Collection will be between 10 a.m. and 1 p.m. I have a little more leniency. I put in 10 a.m. Just as our bottom line, but it can be a little bit before 10 a.m. 9 a.m. is the earliest that you can collect. The reason there's an earliest limit is because those samples can only stay on ice for up to 8 hours in order to still be viable when we are processing them. So the last time that can be collected is 1 p.m. And then the lab part of that day, when we go to process them, we usually start at around 3. And that takes about an hour to process the samples, especially if we have a good team, a good team of 3 or 4 people. Love to have more. That is typically the size that we have in the lab is 3 to 4 people. So if you love learning about lab and doing lab work, it's a pretty easy process. You'll learn on training day, but definitely a big need on those lab days. And then on Fridays, the analysis part will occur from around 12 p.m. to 2 p.m. That can also go fairly quickly. We've gotten samples analyzed within an hour before, but we are going to hopefully be increasing our sample size this year. So it might take a little bit longer, but hopefully not too much longer, if that helps. And then as far as teams assigned to only one location, so teams are it's in the slides. So if you want to, after I tell you, if you want to come back and look at the presentation when I send it out, you can find it in the slides. The way that typically teams are responsible for 2 to 3 locations. I think this year the goal is to have teams responsible for at least or at most 2 locations by us removing Klein's Run. But there still might be 3. 3's the most. And the least would be 1. If you're, I know City Island team is in the past has been responsible for just for 1 location, but that might change for this season too.
And to give you a little bit more there, so any sites that you're going to see on a creek, tributary, we are running 2 locations on that creek to get an upstream site and then a more downstream site. So if you had a question around, is there a team doing 2 different creeks? The answer is no. All teams are typically going to have their own creek. So we have a Peckway Creek team, there's a Conestoga River Creek team or Conestoga River team. So we're typically not crossing between, individual creeks where each teams are managing their own individual smaller creek and watershed. However, though, with the river sites, it may make sense, and we'll want to look at this. You know, like for example, we're going to want to speak with the Harrisburg team to see if there's availability for them to maybe capture the Steelton boat launch location. And if not, that's okay. We can definitely get another team to just do Steelton, and maybe even perhaps getting the further site downstream at Falmouth. That way we can get Falmouth and Steelton done with just one team. But again, we're going to communicate with our Harrisburg team, see if that's okay. And if not, you know, we can run a completely separate team for either just that one Riverside Steelton, and again, another team just for Falmouth. But we will cross that bridge once we get to it based upon your feedback. And we're going to have this out to the public as well. So in addition to you folks that are here tonight, people that aren't here will have the opportunity to sign up for training. They're going to have the opportunity to sign up for locations. So tell your friends, tell your family, if you know anyone who might be interested. Those forums will become public when you get them. So that'll be in the next week.
Any final questions? Well, I think with that, like I said, you'll be receiving an email hopefully by the end of this week. It might be early next week, with all the information that we went over tonight. We'll have a recording link to our YouTube, and then a PDF of this presentation, and then a couple of forms for option for trainings. Thank you everyone for coming out tonight. It's exciting to get a little bit warmer outside and start thinking about this stuff. So yeah, thank you, and have a great night.
Thanks, everyone.
Thank you. Bye.
Bye.